Hello, everyone, and welcome to today's webinar, Deploying SARS-CoV-2 Wastewater Monitoring in Kansas, a case study and adoption of high-throughput automated sample processing. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Ceres Nanosciences. To learn more, visit ceresnano.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Justin Hutchison, Assistant Professor in Environmental Engineering in the Department of Civil, Environmental, and Agricultural Engineering at the University of Kansas. Dr. Hutchison, you may now begin your presentation.
Thanks, Kaylee. As Kaylee mentioned, I'm Justin Hutchison, Assistant Professor here at the University of Kansas. Today, giving a presentation on deploying SARS-CoV-2 wastewater monitoring in Kansas. And so going to be having three objectives that we'll highlight here in a few moments through a case study, and then demonstrating the capabilities that an automated high throughput sample processing has given us the ability to do increased monitoring in Kansas. So our case study is going to be largely focused on Lawrence, Kansas. And so Lawrence, Kansas is a university community with a stable population base of about 100,000, and then plus or minus 30,000 students depending on the season. So as school lets out, those students can go home frequently, and then when we're in session, we can have higher student populations. I introduced this map as kind of a beginning point. So this is case data that we had for Lawrence, and so give you a few moments to think about this as we move through some of the introductory slides, but I'd like to see if you can guess what some of these red intense areas are, which correspond to high COVID-19 cases versus the blue areas, if you think you have some information on or hypothesis of the population targets for these red high COVID-19 cases versus the blue low COVID-19 case areas. But with that, we'll go ahead and get started on the introduction. And so, as many people have probably seen in the news, wastewater-based epidemiology is an emerging method. This has been used prior to the COVID-19 pandemic. The probably largest area of research has been used in illicit drug monitoring. And so there have been lots of studies out there that have made efforts to detect illicit drugs in wastewater to understand potentially use of those drugs within a community. Before the COVID-19 pandemic, there were also early warning disease outbreak indicators. So we've been using enteric virus monitoring in wastewater for a very long time, norovirus measurements, hepatitis A, poliovirus, fairly large study in Israel in a 2018 paper that was looking at a silent endemic of poliovirus. And then kind of the hot topic right now is this idea of using monkeypox monitoring, using wastewater-based epidemiology to monitor monkeypox as kind of this emerges as a concern within communities. And then there have been attempts to do prevalence data monitoring, so to get a sense of the magnitude of the concentrations of the virus in the wastewater, and then again, a little bit of effort in kind of taking that information back. So you have high concentrations of these viruses or maybe low concentrations. Can you take that back to get a sense of how much disease is in a community? One of the limitations of wastewater-based epidemiology is this idea around taking prevalence data, that magnitude of concentration, and determining a specific number of cases. And for the most part, we are not there yet, and it'll be probably a very long time before we can take a concentration that we find in wastewater to use that to predict the number of cases in a community, largely because of the uncertainty on both sides. So if you look at an individual that is shedding SARS-CoV-2, over the span of their illness, that shedding rate or the amount of virus that individual is shedding can span 6 orders of magnitude. And so that 6 orders of magnitude introduces lots of uncertainty when you're trying to take these concentration data and wastewater back to case number. So for the most part, prevalence data, we're doing very good on that, and I'll highlight that in this presentation. But again, I think a limitation, or maybe not a limitation, but something that should be acknowledged, is that taking this prevalence data and connecting it back to the number of cases is something that we're not quite there yet as a field. Wastewater-based epidemiology efforts have grown with national and international coordination. And so you've seen lots of communities jump on board to start monitoring SARS-CoV-2 concentrations in wastewater, lots of dashboards out there. And this is just a dashboard, COVID Poops 19, that's looking at the number of dashboards that are available, universities, countries, and sites that are participating in this monitoring effort. And so you zoom in on Kansas, and right here, the University of Kansas has been leading the wastewater-based epidemiology efforts and monitoring wastewater concentrations throughout the state of Kansas. And so that's something that we started in March 2020 at the beginning of the COVID-19 pandemic, and we have been continuing to date. One of the strengths of wastewater-based epidemiology is understanding trends of disease in a community. And so when we look at what wastewater-based epidemiology efforts can do, it allows us to see those trends and changes in occurrence. And so one of the things that has been mentioned many, many times is this idea of early detection of occurrence. And so when papers were first starting to come out about SARS-CoV-2 monitoring in wastewater, there was lots of effort or lots of research out there that's saying that we were detecting this in the wastewater 5 days before the clinical tests were showing positive cases. And we'll highlight in the data and one of the objectives here that this often has to do with the uncertainty around the clinical case testing. And so when you have these high frequency intense campaigns for clinical testing, the argument is that wastewater monitoring can offer you that same level of information for the community. Assessment of community infection, again, that's one of those ideas. We can track a disease prevalence in a community. So we see an increase in a community, we see it leaving potentially a community, but we really can't tell you the number of cases that, the number of COVID-19 cases that would be in a community. Risk assessment, so risk to utility workers and those exposed to raw sewage. This has been a very hot topic. On the lab side, we tend to treat our samples at a BSL2 plus, almost to a BSL3 level when we're handling these samples, but a lot of the times there is some risk to these potential operators that are collecting those samples for us, and this is something that is an ongoing area of research. And then viral evolution, so source tracking of the virus. This is, again, very difficult. There is some work on cryptic lineages. So this idea of we're observing these SARS-CoV-2 variants in wastewater that don't necessarily have an equivalent in the clinical community. And so it's this idea of where are these variants coming from. But for the most part, what we can do when we look at the identity of variants in wastewater is we compare that to what the clinical community is studying, and what the clinical community has identified are the variants of interest. And so once the clinical community has identified those variants of interest, we often can go back into our wastewater samples and also look to see if we can identify those variants of interest. In general, Kansas is, and this is a good thing, behind the curve in variants emerging within our community. And so on the clinical side, variants of interest are often well established before we see them in Kansas. And so we often have access to those variants of concern in those databases. And so we are often seeing these variants of interest emerge in our wastewater at the same time as the clinical testing side of things. So the big motivation around wastewater-based epidemiology and why these efforts were really undertaken, especially in the early parts of the pandemic, is clinical testing limitations was really driving these pooled testing options. And so when you talk about clinical testing, you need thousands of tests to be able to evaluate a community, whereas in wastewater testing, you only need one wastewater sample. And so most of the times we're collecting a 24-hour composite sample from that wastewater community. And so we can do and process that one sample to get a sense of the community health or the prevalence of COVID-19 within one community. And so these efforts, again, started very early in the 2020, January 2020, even to the end, to some extent at the end of 2019. And then a lot of effort has gone around to reporting these values in a centralized database. And so the CDC has set up a national wastewater surveillance system. And so a lot of utilities, especially in the United States, are reporting this into the central database. Similarly, KU started pool testing, and we started in March 2020. So this was before SARS-CoV-2 and the corresponding COVID-19 disease really impacted our community. So we had a few months of where we had no detection of SARS-CoV-2 concentrations in wastewater. But that also meant that we were very much in the field and detecting this right when this did hit our communities. And so what we did is we could test, and these tests told us if the catchment or that sewer shed, if the SARS-CoV-2 was present, increasing or decreasing in a community. The wastewater-based epidemiology, especially in the early stages of the pandemic, gave us a week's notice when clinical testing was limited. It helped our Kansas communities prepare for spikes, especially in our rural communities, where both clinical testing was not very prevalent, and also general health community resources may be more limited in rural communities. And so when we were doing this monitoring, we were working closely with our state regulator, who was then passing the information on to the regional county health departments. Wastewater-based epidemiology showed concentration of virus in the sample, but again, we weren't and cannot still tell people the specific number of cases. And again, lots of headlines out there. And so if you do some Google searches, you can find that we've been doing this again for quite a while. And there was lots of news interest in these developing efforts. I think one of the things that's important to highlight when if someone's interested in launching these is that our communication strategies and how we communicated our results from our wastewater-based epidemiology efforts really evolved over these last 2+ years. And so when we were in our pilot phase, which was, can we do this? Are we interested in doing this? We were working closely with the city of Lawrence, which is the hometown of the University of Kansas. And so the city of Lawrence was really great. They were providing us samples. We brought those samples back to our labs, concentrated the virus, extracted the RNA, and then quantified. And then we reported these informations with a close partner at the Kansas Department of Health and Environment. After our kind of pilot phase testing, we started expanding this to more of a statewide testing phase. And so again, this kind of took on a very similar structure where local municipalities were sending us the samples, which is highlighted in the blue arrow. We were extracting the virus, quantifying the virus, and data processing here at KU. And then we were reporting the data. And in general, we kind of took the philosophy of giving the data out to as many people as possible. And so we always reported our data back to the local municipalities. If we had the direct connection to the local county health departments, we sent that data directly to them. And then again, we sent this information on to our state regulator, the Kansas Department of Health and Environment, who then would also follow up with local county health departments. And so again, kind of a philosophy based, we wanted to get this data out to as many people, and really we wanted to get the data out to anybody that was interested in seeing this. And then for the most part, the dashboards, which have become pretty common and popular, we mostly relied on local municipalities showing interest and if they wanted to report and develop a dashboard. And so Lawrence developed a dashboard, which is still live, and the link is there if you're interested in seeing that. As we kind of enter what I think of as more of a mature phase of our wastewater-based epidemiology efforts, this got even more complicated. And so again, we brought on another entity, the Kansas Health and Environmental Laboratories, which are the labs that are for the Kansas Department of Health and Environment. And so we started splitting samples since we were monitoring many counties in the state of Kansas. And so again, municipalities are still sending us samples. We split those based on the labs. The labs are responsible for quantification and data processing, and then the reporting also got a little more complicated. And so at the University of Kansas, we're still reporting to the local municipalities and sending this information on to the Kansas Department of Health and Environment. Part of my learning process is realizing that our Kansas Department of Health and Environment kind of is a little bit of two entities where you have the health side and you have the environment side. And so a little bit more communication had to occur between our environment and our health side. And so that's fairly underway and fairly established. And so then our health side is reporting our concentrations to the CDC through that national wastewater surveillance system. And again, also disseminating to these local county health departments. So again, I think an important point to make for this is that these communication strategies have really evolved over the process. And again, we largely took the philosophy of trying to get this information out to as many people as possible. So I mentioned objectives. 3 objectives that I want to highlight for this talk is identify potential benefits of SARS-CoV-2 wastewater surveillance as an early warning indicator of an outbreak in a community during infrequent clinical testing periods. Provide evidence that suggests SARS-CoV-2 wastewater measurements offer similar community-level health information as high-frequency clinical testing. And then last, recognize how an automated workflow can increase capacity and reduce total processing time. Right, so for the first 2 objectives, we're going to use a case study, and for that first objective specifically, where we're looking at wastewater surveillance as an early warning indicator. So again, we're in Lawrence, Kansas. We collected this data from June to December 2020, and so this is about a 6-month window where we're presenting data from the case study. In this particular community, we have 2 sewer sheds. So on the bottom left, we have what we refer to as the Wakarusa sewer shed, and then kind of the right top is the Kansas sewer shed. And these sewer sheds mostly flow into the respective wastewater treatment plants. And I say mostly because Wakarusa, the sewer shed actually flows to both plants. And so realizing those differentiating flow patterns was really important to account for when we were looking at our SARS-CoV-2 concentrations and making that connection back to the case data that we were getting. So again, counted for that wastewater flow as the Wakarusa sewer shed flowed to both utilities. Another concept that I'll highlight is the wastewater strength. And so wastewater can be diluted, especially when you have rain events. And so we wanted to account for that varying wastewater strength using normalizing biomarkers. And then last, COVID-19 cases were geocoded by location within the 2 sewer sheds. And this data was obtained from the Douglas County Health Department. And I mentioned earlier in the beginning of this presentation, the red versus the blue. And so red is high case data. So this is the COVID-19 cases, and blue is lower case counts. And in general, we found that the red intense areas corresponded to high student density populations. And we confirmed what we are observing with the geocoded case data by going into the sewer system. And so this is kind of sub-sewer shed testing, popped open a manhole, grabbed some samples that were specific to these communities, maybe in the higher intense red versus the blue, and observed in our wastewater that this was also true, that our wastewater from these student communities tended to be quite higher than communities that might be, for example, single family residence home, more in these kind of blue areas that you see on this map. So when you look at wastewater flow, it's important to realize that there's going to be some variation. And so if you're doing a grab sample, it's important that you realize if you take a grab sample at maybe a low flow event, where maybe not too many people are showering or flushing their toilets, versus a high flow event where you might have lots of people showering or lots of people flushing their toilets. So for example, in the early morning when people are waking up. It's also important that you distinguish whether you're a weekday or a weekend. So some of our communities, just because of limitations, do have to do grab samples. But in Lawrence, we did 24-hour composite samples. And so we collected a sample of wastewater that was representative of that 24-hour period. However, it's also still important to note that you can get variations in flow from day to day. And so we saw that variation in flow within a day, mostly just because of human patterns, but you can also get variation in flow because of rain events, for example. And so infrastructure systems are not perfect. They can be leaky. And so when you get rain events, you get infiltration and infusion, so that water coming from outside into the pipe, which can dilute your wastewater. And we also see this in the Lawrence wastewater treatment plants. And so here again, when you look at the Kansas wastewater treatment plant, here we're about 1 million gallons per hour. And then when we have these large rain events, the one can jump by a factor of 2.5 to 2.5 million gallons per hour. And so again, these large flow events can be indicative of diluting your wastewater, which is something that's important to account for if you want to start comparing, for example, between days and weeks. I mentioned Wakarusa is a little bit different. The Wakarusa treatment plant is flow limited. And so when we have these really high excess flow, the Wakarusa plant will divert some of its flow to the Kansas. So to account for that variation in the wastewater strength, we took 4 measurements: biological oxygen demand. So this is a readily available, regularly measured biomarker that we can use to normalize our SARS-CoV-2 concentrations. Ammonium, also readily available, regularly measured in wastewater treatment plants. The next 2 we measured more in the laboratory, the university laboratory. Creatinine, it's indicative of human activity. And the reason why we selected creatinine is that it has a similar half-life to SARS-CoV-2 in a wastewater distribution or collection system. And so this biomarker could be important to include so that you understand SARS-CoV-2 virus decay in that collection system. And so if you have a really big collection system and the wastewater sits in there for a very long time, that SARS-CoV-2 virus has more time to decay in the system. However, creatinine is excreted in urine, and so that's a bit different than the SARS-CoV-2 virus, which is excreted in feces. So we also monitored mitochondrial DNA. And again, the idea there for selecting that was excreted in human feces. Now, the much more popular common method for normalization is the peppered mild mottled virus, or PMMOV, which is an RNA-based virus that's also excreted in human feces. And so we also now measure that, but in these earlier stages of the pandemic, we did a fairly large comparison of 4 different biomarkers to account for that variation in wastewater strength. Lots of methods are out there to concentrate that SARS-CoV-2 virus. In particular, for this study, we were using a 12% PEG solution followed by extraction. At the beginning of the pandemic, you had electronegative membranes, ultrafiltration, and general centrifugation steps. We tested all 4 of these, and in general, our recoveries were best with the PEG precipitation. And so we used a 12% PEG solution with a bovine serum albumin that improved our recoveries. Our recoveries, which were measured with a surrogate virus, Murine Hepatitis Virus, could be upwards of 32%. However, it's important to note that viruses, SARS-CoV-2 and MHV, like to partition between different phases. And so when you have a solid sample, or you have solids in your wastewater sample, we often found that both the MHV, or surrogate, and SARS-CoV-2 like to partition into those solids. And so this is, again, maybe one of those reasons why understanding how people process their samples is really important, because if people excluded the solids, they may be excluding a large portion of their viral measurements in their sample. And so if that lab consistently excluded their solids, in general, I would say their data is good for observing those trends. But if you were to compare their sample to a sample or a lab where they included the solids in their sample, you would get very different measurements. So that virus concentration and RNA extraction, virus concentration, we started with 36.5 mils of wastewater. So after concentration, we got that down and we extracted down to 60 microliters of RNA extract. And then our RNA quantification was predominantly or largely done with droplet digital polymerase chain reaction. We use droplet digital polymerase chain reaction, technically a little bit more difficult than standard PCR or qPCR. We had a little bit better response with ddPCR with avoiding inhibition from the wastewater sample. So wastewater samples are are complex samples, and there can be compounds that interfere with your quantification for PCR. And we, again, tend to have better luck in our lab when we were using ddPCR. So our quantitative limit of detection for this process, we were measuring both N1 and N2, and I'll highlight what those are here in a second. But our quantification limit of detection was 1.28 gene copies per milliliter of wastewater. And then for N2 was slightly better at 1.1 gene copies per milliliter of wastewater. I mentioned quantification, and so we use the CDC SARS-CoV-2 primers, which have been pretty standard for general qPCR, ddPCR measurements in these wastewater-based epidemiology efforts. And so we use the TACMAN probe in our ddPCR instruments. So the primers are available, and happy to answer any questions around this more specifically if anybody has those. But again, this is a largely established, well-established protocol that the wastewater community has been using consistently. So I mentioned that we partnered with the Douglas County Health Department, and they were the ones responsible for providing the case data during our comparison period. And so again, we had our Wakarusa sewer shed and our Kansas sewer shed. So it was really important that we didn't just get positive case counts for Lawrence. We had to understand where in the sewer sheds people were contributing to their feces or their fecal matter into the wastewater and which sewer shed. And so the assumption here was largely that if an individual tested positive, they would be at their primary residence. And so that's what you have here is that geocoded positive cases based on residence. And again, the blue would indicate 0 or very low COVID-19 cases. The red to orange would indicate high COVID-19 cases. And something to acknowledge as a limitation is that, so we assumed a primary residence, but again, Lawrence Memorial Hospital was located in the Kansas sewer shed. So if you had hospitalized individuals, they would likely be contributing to the Kansas sewer shed, even if they had, for example, been living in the Wakarusa sewer shed. And so collecting that case data, again, can be often very noisy. And so you can use some smoothing curves to understand general trends in that case data. And so here we have daily new COVID-19 cases on the left or y-axis over the time period of our study, again, from June to December 2020. And so in general, we observed during this period 3 large case spikes for our community. So taking that case data and plotting that against our SARS-CoV-2 concentrations, and again, I'm in this case reporting N1 concentrations on the y-axis, and in this case only presenting the data where we normalize to mitochondria or creatinine, those human-specific biomarkers that would be found in the wastewater. And in the combined sewer sheds, so we're looking at the combined data for both sewer sheds, we see our blue case data trend, those 3 spikes. And then in our wastewater data, we see our 3 spikes when we normalize to creatinine. And in the bottom graph, we see in orange our SARS-CoV-2 N1 concentrations when normalized to creatinine. And again, I always kind of joke, you got to cross your eyes to kind of see the trends, but there are 3 spikes in this data. And I'll follow up with some of the statistical analysis that were performed so to kind of convince you that these 3 spikes corresponded to that case data. We could also divide these out by sewer shed. And so in the Kansas sewer shed, we saw the the creatinine, that top middle graph. We see those 3 spikes of N1 concentration. And when normalized to creatinine, again, those 3 spikes. The bottom graph is mitochondria, those 3 spikes. And then in the Wakarusa sewer shed, where the data was a little bit more noisy and a little bit more difficult to interpret just visually, but the Wakarusa sewer shed also saw some spikes in the N1 concentration. So some of the statistical methods that you can use to evaluate the quality and the correlations of your data, in this case, we used Spearman's correlation data. And so a positive correlation here being all the way up to +1. If you saw an increase in the SARS-CoV-2 concentration in the wastewater, you would also see, or you correlated to an increase in the COVID-19 cases. A negative correlation, -1, would be a SARS-CoV-2 increase in the wastewater, but then you saw a decrease in the number of COVID-19 cases. So in general, when we're looking at correlations between SARS-CoV-2 concentration in the wastewater and COVID-19 cases, we're really wanting to see that +1, or that strong correlation between our measurements. And what we observed in the combined sewer shed when we looked at 21-day cumulative cases is that our wastewater measurements were detecting SARS-CoV-2 N1 concentrations at the strongest correlation 5 and 6 days ahead of the case data and the COVID-19 case data. And again, our strongest correlations were observed when we normalized our concentrations to those human-based biomarkers. So again, everybody's contributing to the sewer shed, this creatinine and this mitochondria. And so these normalizations give us a sense of that strength of that wastewater in a system. So this trend, or this observation of this 5 or 6 day detection of SARS-CoV-2 concentration in wastewater versus case data was observed very early on in these efforts. And so here, again, in a paper 2020, where they were looking at SARS-CoV-2 concentrations in primary sludge, red, again, they were detecting SARS-CoV-2 concentrations about 5 days before the corresponding case data. And again, these trended very closely with that 5-day gap. However, in the earlier stages of that pandemic, the case clinical testing was very sparse. It was hard to find, it was hard to get. You had to have very clear indications that you were sick before you could even get testing. However, at the University of Kansas, when we wanted to try and bring students back in the fall of 2020, we instituted these high-frequency clinical testing campaigns with very high turnarounds. And so when we look at more this fall time period where students are coming back, I would say that we had probably greater quality or better quality COVID-19 case data with respect to both timing and just the actual number of individuals that were sick or testing positive, even if they were asymptomatic within the community. And so across the board, you saw our correlations improve. So we saw much stronger correlations with our SARS-CoV-2 wastewater concentrations versus that case data. Both for both N1 and N2, and again, across all of these biomarkers that we used. But again, we're still seeing stronger correlations with our human-specific creatinine and mitochondria. And again, more importantly, we saw that our correlations didn't have a daily offset, which means or suggests that our SARS-CoV-2 concentration wastewater measurements were giving us a similar community level health information as these intense high turnaround or fast turnaround clinical testing campaigns that we were using in Lawrence, Kansas. Now, the thing that scientists always love to say is correlation is not causation. And so just because these are correlated doesn't necessarily mean that a sick individual, the more individuals that are have COVID-19 in a sewer shed doesn't necessarily mean that it's causing a higher or an increased SARS-CoV-2 concentration in your wastewater. And so we used a slightly different statistical method called Granger causality, which is evaluating the ability, in this case, of our SARS-CoV-2 wastewater concentrations to predict the number of COVID-19 cases within a community. And so this is not taking a concentration and telling you the number of cases, but it's more looking at trends. As we see our historical trends of SARS-CoV-2 concentrations in the wastewater, do we see the same trends in our COVID-19 case data? And in general, this is a chord diagram. What we have is, again, our normalized SARS-CoV-2 N1 concentrations to mitochondria and creatinine did have a causal relationship with those case data with a 1-day or 4-day lag based on this evaluation matrix. And so again, this is suggesting that when you have more individuals, more COVID-19 cases in a community, that you're seeing an increase in that SARS-CoV-2 concentration, and that those SARS-CoV-2 concentration wastewater measurements could be used within one community to potentially predict the number of COVID-19 cases. So again, this is important that I keep saying one community because this relationship was developed based on our SARS-CoV-2 concentrations in wastewater. And so if you were to go to a different lab who used maybe a slightly different method, they might get a different report on the number or that concentration of SARS-CoV-2. And so you can't necessarily take that concentration and divide it by some number and get the actual number of COVID-19 cases. Right. So key research outcomes from the case study. So we observed strong correlations with human-specific biomarkers. So again, normalizing can play in a really important role in understanding those trends. We use creatinine in mitochondria. A very popular one now is that PMMOV, pepper mild model virus. Both biomarkers provide opportunities to account for SARS-CoV-2 characteristics in the wastewater system. So again, creatinine was selected because it has a similar half-life in collection systems as SARS-CoV-2. Mitochondria was selected because it represents that solids partitioning that SARS-CoV-2 might exhibit. And then Granger causality tests indicate normalized SARS-CoV-2 wastewater concentrations may predict that 21-day cumulative cases. Switching to our last objective, recognizing how an automated workflow can increase capacity and reduce total processing time. So Cirrus Nanosciences was developing these nanotrap magnetic virus particles that could concentrate the virus and then follow that into the extraction step. And so what we transitioned to was largely manpower-driven PEG extraction, a PEG concentration RNA extraction step. And we combined or modified these two steps into an automated platform. And so again, virus concentration was achieved by these nanotrap magnetic virus particles, and then RNA extraction by a MagMex microbiome ultra kit, which could be automated in a Kingfisher platform. And so these are magnetic bead-based platforms that could improve and significantly improved, as I'll show, processing time in labs. Before we adopted the platform, though, we wanted to evaluate the effectiveness. So just like when we evaluated the 4 initial protocols, we also evaluated this automated platform using or against our PEG. And so here on the y-axis, you have percent recovery of our surrogate, Murine Hepatitis Virus. And then on the x-axis, you have these communities that we've been testing in the state of Kansas. And the first thing that you'll notice is that the nanotrap without the enhancement reagent 1, and even with the enhancement reagent 1, we had much higher recoveries of our MHV virus compared to our PEG method. And so again, this was part of this narrative is accounting for that solids. And so MHV, when we account for both the solids and the liquid phase of wastewater, our MHV recovery and PEG method could be much higher in the 50%. But with this automated platform, we didn't have to do that 2-stage recovery. And so if we were only looking at the liquid phase, the nanotrap showed much higher recoveries of our surrogates. There were cases still where some utilities, we just sublet, for example, we were never able, there's something in that wastewater that's very interesting. We were never able to recover neither our MHV surrogate nor the SARS-CoV-2 in those samples. Looking at a comparison of CT, so this is that threshold level by which you detect N1 concentrations, and in this case, qPCR. So the lower the number, the quicker you might detect N1 concentrations in your wastewater sample. And again, the general trend is that the red squares are higher, which is the automated platform magnetic nanotrap particles with the enhancement reagent 1, blue being in general, some of these that are similar to our original PEG methods, and that would be the no enhancement reagent 1. And then we have an X, our PEG method. So again, we were seeing both enhanced recovery of our surrogate virus as well as earlier detection or lower CT value for detection of N1 in our qPCR samples. To kind of give you a sense of the amount of time saved, I kind of prepared this table if we were looking at processing 24 samples. And so we have these procedural steps of preparation, virus concentration, and viral RNA extraction. So these are kind of the steps before the virus quantification. And our preparation, 12% PEG is very difficult to make and is very viscous, and so it's also very difficult to handle. And so preparation was much higher, about 4 hours with 2 undergraduate students and a technician. Whereas in the automated platform, we have a much shorter time, and the reagents are much easier to work with. They're not nearly as viscous as that 12% PEG. Virus concentration, 3 hours, 1 undergraduate, 1 technician. Largely, a lot of this time is focused on centrifugation. So for PEG, there's a 2-hour centrifugation step that was required. Now for virus concentration, it's mostly 1 technician, and this is mostly runtime on the automated platform. Virus RNA extraction, again, when we were doing the 12% PEG method, we had to transfer the concentrated viral particles into our RNA extraction. And again, this was about 3 hours, 1 undergraduate or technician. And then now on this automated platform, we're at 1 hour. And so if you're looking at just total processing time to get 24 samples, in our lab, the PEG method would take us 10 hours and a combination of at least 2 undergraduate students and a technician, whereas now we're at 4 hours and mostly working with a technician, but also an undergraduate student helping sample preparation or help process preparation. And so again, this automated platform allowed us to expand our testing throughout the state of Kansas. And so this is, the red dots are just showing higher concentrations. And we expanded this testing for 95 counties in the state of Kansas. And one of the important things that also let us do is that this testing, when we first started, was mostly quarterly. And I'll show that here in a few moments that if you really want to detect these trends, you've got to be much more frequent than that. And so the automated platform allowed us to move to at least biweekly testing, if not more frequent. And to kind of show you what that data looks like, I'll highlight 6 communities that we're evaluating. So a wastewater facility in Johnson County, Hutchison, Kansas, Concordia, Hayes, Colby, and Wichita. So these are all, again, municipalities in Kansas. And so when we've moved to more to that high frequency testing, you can start to see these trends and that N1 SARS-CoV-2 concentration in these samples. And again, you need that kind of more high resolution, that kind of weekly or biweekly sampling to catch these spikes in that N1 concentration that you observe in all of these communities. Whereas if we were still just testing quarterly, we'd miss that. We also we're able to start sequencing. And so we are doing sequencing now with Arctic version 3 primers, 75, 76 alternative, and 77. And we're running MySeq pooled 3-month sample runs. And we started this in 2021. And so some of our first data here is for October, November, December 2021. And for the most part, when you look at October in Kansas, we're largely seeing, these kind of what I refer to as these legacy variants. So COVID-19, some of the variants that were introduced in 2020. In November, we start to see or detect Delta, and then really in December of 2021 is where we start to see that Omicron pop up in this particular Lawrence community. If we look at all 13 Kansas communities, this was similar trends. For the most part, when we were looking at October, the percent variance in these communities were mostly those legacy 2019, 2020 variants. In some of the communities, we did see Delta. And it really wasn't until December 2021 where we start to see Omicron pop up in some of these communities. And the encouraging thing about this wastewater sequencing data is that we detected Omicron occurring or popping up at the same time as our clinical testing. And so if we look at the clinical testing data across the state, most of what was being detected in October was Delta, all the way up until December, where then in December you started seeing Omicron pop up in these clinical testing cases. Again, always the interesting thing is why do we see some of the 1920 variants in our wastewater that are predominant versus the clinical testing, which is mostly showing Delta and then later on a little bit of Omicron, is still a very interesting question that we're pursuing. So summary from this presentation, again, from the case study, strong correlations were observed with human-specific biomarkers, creatinine and mitochondria. Both biomarkers provide opportunities to account for SARS-CoV-2 characteristics in that wastewater system, so either half-life or solids partitioning. Again, Granger causality tests indicate normalized SARS-CoV-2 wastewater concentrations may predict our 21-day cumulative cases. And then for us, the automated platform enabled higher frequency statewide testing and variant tracking via sequencing. Just like to take a moment, this has been a very large group effort. Professor Belinda Sturm and I helped get this off the ground in early 2020. Lots of undergraduate students and technicians have been helping with this process. And so just take a moment to acknowledge the students and coworkers that have been involved. Also, a lot of the cities who partnered with us on the early stages of this, City of Lawrence, Johnson County, and Olathe. Also like to take a moment to acknowledge funding sources. So Kansas Department of Health and Environment, Patterson Foundation, Sears Nanosciences, University of Kansas Chemical Biology and Infectious Disease, University of Kansas Center for Molecular Analysis of Disease Pathways. Sequencing was provided by the University of Kansas Genome Sequencing Corps, and then financial support also provided by the National Institute of Health under grant number listed there. And with that, happy to take any questions. I left some of the conclusions up as people formulate questions, and like I mentioned, I'm happy to take any questions.
Thank you, Dr. Hutchison, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, let's get started. Our first question asks and mentions, it's impressive that you built this program from the ground up. What are your top 3 learnings that you would share with a lab just starting this path of surveillance?
Yeah, so thanks for the great question. I think kind of the 3 things I would strongly encourage someone if they're just starting this out is first operator relationships, municipal relationships. So we really would not have been able to do this work if we didn't have all of these operators in Kansas sending these wastewater samples through the mail. I'm sure I probably also have to thank shipping entities for, probably not questioning too much that we're shipping a bunch of wastewater across the United States. And I'm sure the massive amount of increase in the number of wastewater samples that were being shipped across the United States was very large. Again, still building that community, again, leverage any networks that already integrate with operators in your, in this case, in the United States. And so we have state-based professional societies. We were also able to leverage to help connect us to the municipalities for this effort. And then also kind of a 2-part understanding the limitations of wastewater surveillance. So it took a lot of work for us to be able to take our wastewater concentrations that we were measuring in one community and connect those to the case data. And so that took a lot of work, but then also bringing on those people. So again, the local county health departments in our case, bringing those people on board to be willing to give us the data, to help us interpret the data on the case data side, because, I think we tend to focus that there's a lot of uncertainty on the wastewater side, but I think there's also a lot of uncertainty on the clinical testing side of things. And especially now that we entered the stage of the pandemic where not all, but a lot of testing in the United States are people testing at home, and they don't always report if they have a positive case or not. Thank you for that question.
Great, thank you. Next question here. What is next for Kansas for wastewater testing?
It's always an interesting question. So we've been working on kind of 1-year timelines, and our timeline for this round of testing is coming up in June 30th. And so we're still trying to figure out how we're and who is going to do this. But I think the important and kind of cool thing about this wastewater-based epidemiology efforts is this idea that this is really kind of a very powerful community health tool that a lot of health departments are using now in their toolbox. And so there has been a wide variety of interest across the state to continue these efforts. And again, that June 30th, there may be some transition. At the university side of things, we just have an NSF project launching called Adaptive and Resilient Infrastructures Driven by Social Equity. And so a lot of these concepts will be used in assuring that collection systems, especially in invested communities, are updated so that those wastewater collection systems can cause leakages, for example, into other communities or into drinking water systems. So these are combined sewer overflows and sanitary sewer overflows. I should pause and back up a sec. So because we have aged infrastructure in a lot of communities, when it rains or even just when there are increased amounts of sewage flow in a collection system, this can sometimes overload the treatment systems. And there's these raw sewage discharges. These raw sewage discharges can affect communities both locally if you had an infrastructure breakage within the community, or in downstream communities, for example, on the Kansas River, where an upstream community releases and then it impacts the downstream. And so a lot of these, again, expanding beyond SARS-CoV-2, a lot of these tools are usable in other infectious disease. And so we'll be using these tools to both monitor and then also suggest improvements in infrastructure systems across the state of Kansas.
Great, thank you. Next question, how effective were the tests, and were or will those results be available to the community, and where would we be able to find these results online?
So we did publish at least the case data set in FEM's article. I think the slides are going to be released after this presentation. And so there's a citation for that has my last name, Hutchison. And so the Lawrence case data is already out there on the internet. So if you can just search Google Hutchison SARS-CoV-2, that article will probably pop up. And all of the corresponding underlying data is available on Mendeley data. So we, in addition to the publication, we shared all of our raw data as a data publication.
Another question here, what type of analyzer are you using?
I'm sorry, could you repeat the question?
Yes, what type of analyzer are you using?
So this is a multi-layered question. I think it depends on which specific aspect of the testing that you're asking about. So for the concentration and RNA extraction, we're now using the Series Nanoscience Nanotrap beads, particles, the MagMax RNA extraction, and the Kingfisher platform. And then that transitions once you start doing the quantification, the Bio-Rad droplet digital PCR system. And then for sequencing, we're using the MySeq run.
Great, thank you. Our next question here asks, can you list your LOD again?
Yes, I write these down because I am very particularly bad at memorizing this. So the N1 quantitative limit of detection was 1.28 gene copies per milliliter of wastewater, and for N2, it's 1.1 gene copies per milliliter of wastewater.
Great. We have time for a few more questions here. This next one asks, what are your methods for normalizing for creatinine and mitochondria?
So for creatinine, we used an ELISA test. So this is just a colorimetric intensity test based on the concentration of creatinine in the sample. And then for mitochondrial DNA, we developed a qPCR assay, and the primers, and again, all of that information is also located in the publication.
Thank you. Next question. When using the liquid phase only for nanotrap beads, do you pre-process the sample in any way in an effort to release additional virus particles from the solid phase first?
So the only pre-processing step that we have right now is that enhancement reagent 1. So we do add that, which again, in our assessment showed an increased recovery of our MHV, and also across the majority of our participating municipalities showed an increase in the N1 concentrations, or ability to detect the N1 concentrations in our samples. With the transition from the platform, we stopped pre-centrifugation. So we are also now including the solids in our wastewater samples. So again, there's kind of a transition point between objective 1 and 2 and objective 3. So in objective 1 and 2, we did pre-centrifuge to separate our solids for the PEG method. But then after and once we transition to the other platform, we stopped doing the pre-centrifugation step.
Great. Well, it looks like we have time for one more question, so we'll go ahead and wrap up with this one. And it asks, for your creatinine, etc. correlations, you used your raw gene copies of virus per volume of sewage, CDC and majority of dashboards are normalizing the N concentrations to the total daily influent flow and then sewer shed population. Do you think this is the correct data to use for those normalization correlations with case data then?
So that is an excellent question, and you're not going to like me because I'm not going to say there's a right way yet. I think there's a lot of work out there trying to figure this out. We generally still report concentration data for most of our municipalities. The reason why we do concentration data when we're just directly reporting to our municipalities is some of them have lagoon systems. And so in those lagoon systems, it's really hard to get a sense of the flow in and out of those lagoon systems. However, when we're analyzing the data, we do take into account the flow data. And then that is also taken into account when we use those normalizing biomarkers. We also have looked at potentially normalizing to population. But when we were looking at Lawrence again, we tended to avoid that for two reasons. We had some pretty large shifts in population over the course of the pandemic. And so how did we account for that large shift in population was really difficult. The other thing to be aware of is depending on how you're collecting your sample, the number of people contributing to a collection system varies. So for example, this is more anecdotal from my children, but my children tend not to like to poop at school. And so they'll wait. They won't poop during the day at school. They'll wait till they go home. And so again, you have these discrepancies of when people are using the wastewater collection system. And so those can be hard to account for when you talk about just a population number. And so that's why we tended to prefer those human-specific normalizing biomarkers that we actually measured in the wastewater. We did try to take back the biomarker concentration into an actual population number. The creatinine showed that we were generally about an order of magnitude population estimate lower, which would suggest that creatinine is probably degrading in the system. And likewise, SARS-CoV-2 would be degrading in that collection system. The mitochondria normalization needs more work. There have been efforts to quantify the number of mitochondria that an individual sheds, and there have been efforts to quantify mitochondria in wastewater collected at the municipality. But when you try to back calculate the number of individuals based on those numbers, we ended up estimating that like Lawrence had a population of like 1 million, which is much higher than what we actually have in Lawrence. And so there's still some work on these normalizing biomarkers if you want to try and get a sense of the exact number of people contributing to that system.
Great. Well, thank you again, Dr. Hutchison, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Ceres Nanosciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.